in weddings, we always have recognize the person that has come from the furthest away. So we're going to recognize Dr. Aqeilan as the one who's come from the furthest away, Israel. Thank you very much for making the trek here. Rami is a former apoptosis scientist who has pivoted to neurodegeneration, and he's going to follow on AAV. Thanks very much, Rami.
Thank you, Alex. Yes, and I'm really very happy to be here, as everybody else. And I want to thank Maura and Sabrina for engaging me and bringing me over. It's really a pleasure. And thanks for all the speakers for making a wonderful introduction. So you already, everybody studied 101 biology, 201. So now we're going to a higher level. So I'm not explaining anything like the young kids. And remember, there's an exam. And actually, I want to start with the exam and share with you some facts and numbers, which I like to start at the beginning, just to give you an idea of what we are dealing with. So, true or false? Okay, so I have to use this thing. Okay. There are over 7,000 rare diseases, with more being discovered every day. Excellent, class. In the US, a condition is considered rare if, oops. Okay, sorry, I have to remember to coordinate, because I didn't want to do this in my, a condition is considered rare if it affects fewer than how many persons combined in a particular disease group? A, B, C, D. C. Excellent. B. So, for Alex, excellent. What can I tell you? Yeah, but we are in America. Sorry. Rare diseases affect how many million people worldwide? Alex, stay silent. So it's D, right? 350 million people worldwide are affected by rare diseases. If all the people with rare diseases lived in one country, okay, it would be the world's 6th most populous country. True or false? Who said false? Alex, good job. It's the 3rd most populous, actually, not the 6th. Only 30% of rare diseases have an FDA-approved drug treatment. True or false? False. What is the percentage? Only 5%, right? Very sad. Approximately 50% of the people affected by rare diseases are children. True? Good job. Rare diseases are responsible for 35% of death in the first year of life. True? On average, it takes most rare disease patients 5 years to receive an accurate diagnosis. Well, the statistics says it's 7 to 8 years, unfortunately, but I think we are approaching there. So we are on right track with all the advances and access to health systems in the US in particular. So 30% of children with rare disease will not live to see their 10th birthday. True or false? This is false. 5th birthday. Many diseases with neurodevelopmental diseases, they will be very harsh. I work on a syndrome called Worry syndrome, WOX-related epileptic encephalopathy, and most patients would die before the age of 4 years. Okay? And there is a genotype-phenotype correlation, which is really could affect these diseases and others. Okay, so this is the reality. These facts could be difficult, right? But they highlight a very critical thing, which is that rare is not really rare if we look at rare diseases altogether. So our mission is really to understand these diseases more in depth and to really provide therapeutics, diagnostics, and to help the patients and the families. So during the last 5-6 years, I'm a cancer biologist in training, so this is a consent. And since, I would say more times it flies, 2016, we started working on neurodevelopmental diseases. And since then, we started with one gene, the WOX gene, but now we work on several others, FAM, DLG4, CTNNB1, and so on. And the beauty of these genes is that, unlike cancer, which is a polygenic disease caused by many genes, these are mostly monogenic. And I never thought about the brain before. Why the brain? Nobody, it's not an important organ. So we did not work on it. But now, when I saw how these monogenic diseases have this potential of being corrected with, for example, gene therapy, that was really fascinating for at least a researcher to see that this is possible, okay? And this is why we started working on several of these genes, but today we will be talking mainly on DLG4, right? And I'm gonna skip a lot of the introduction because of all the previous speakers and just focus that on a few things just to remind you that will serve my studies. So you all know these clinical manifestations that come with the disease. And the research goal that we set to ourselves is basically two very important things. One thing is to model the disease, to look into the molecular mechanism of how is this affected, and then to develop therapeutics based on this, to translate the knowledge of this basic understanding into something that we can really help the children with. So this is very easy, and this is our gene. You all know it, the chromosome, the protein that encodes. It's a postsynaptic protein. It's a scaffold. So it interacts, it partners with so many proteins and bring them together. So it just holds everybody together. It's just this pillar that brings every other protein at this very particular location in order to allow for the electricity, the electrical activity of this neuron to pass and to be coordinated and to make this network. So everything works in synchronization. Everything works in harmony, okay? What is a few facts about DLG4? It's really very low during brain development. So very early during brain development, it's very low, and it just starts to go up during later stages of brain development and in adulthood, which makes sense, right? During brain development, very early on, there's just the birth, as Dr. Motri described. So this the birth of the neurons, but then they start to make these networks later on, right? They mature. It takes them time, and we wait 9 months, and then, right, this is what we call in our textbook neurogenesis and maturations of the neuron. It is even more complicated when we looked at regulation of the mRNA, because what you see here is that there is very specific and tight regulation on the RNA of DLG4, and it has been suggested that these two genes, the PTP1 and 2, would regulate post-transcriptionally the splicing of DLG4, and this is how its expression is regulated, at least in the mouse. So it is tight, it is complicated, and this is why I was saying that RNA is not the whole story, and we need to look at the protein, which is the most important functional tool at least in the neurons. We're talking about these synaptic densities, and you saw this several times. I just remind you, we're talking about a protein that is bringing all these different proteins together and make this scaffold function. And it is composed of these different domains. You saw this before. Very important domains that will become relevant later on. And this is the main functions. Okay? So it is the most abundant. Okay? It's a very important protein. And it is responsible for synaptic plasticity that is happening at these edges of the neuron. And it controls the postsynaptic densities and the dendritic spines, which is allowing this networking between the neurons. And it is also important for this multi-innervated spine formation, which again feeds on into how the signal is moving from one neuron to another, how the neurons are really networking together. And these are the outcomes in the Shine syndrome you all are aware of, and we'll try to address some of these in our strategy of how to overcome this deficit or this defect. I think it was 2022 when we started this project, or 23, I don't remember. And we received these 3 IPS lines, 2 from Chio, and 1 from Harvey, from Jackie. And as you can see here, these are the protein chains, these are the mutations. And we have 1 missense, nonsense, and frame shift. Okay? And they are affecting these different domains that I showed before. So it's kind of, we wanted something to be not only 1, but also more, although 1 is still important, and I emphasize. But we wanted to cover different sides of the gene and see if there are differences. And as you will see, there are differences. What they are, stay here, don't go anywhere. No, you will learn. So what we did, and when I say we, this is Dania in the lab, first year PhD student. She got these iPSCs, and now we wanted to produce neurons during this very established protocol, the NGN2, excitatory neurons, or cerebral organoids, unguided organoids, little bit different what Dr. Motri used, but pretty much the same principle, and study them at the molecular function, determine loss of function, gain of function, synaptic strength, and then AAV, what you heard before. These are the 2D, these are the 3D organoids. Everybody covered this, so I just don't want to be so much redundant. iPSCs, these induced pluripotent stem cells, you can make any cell type in the body, right? Any cell type, different protocols. So we are interested in these neurons, and then you can do whatever studies of them with the MIA, with the RNA-seq, the single cell, study them by imaging. We can do a lot of things in there. And this is the cerebral organoid protocol we did. Very same principle of what was presented in the previous talk. Before moving on, I just want to also tell you what is really gene therapy about. We want to use DLG as the gene, as the medicine that we are giving to the patients or to the kids, okay? So basically, we are replacing or adding this gene to this affected cell that is harboring this mutated gene, okay? So the gene is the drug. The gene is the medicine we are giving to the children, and it is given by different routes. It could be injection in the blood, it could be injection in the brain. Mostly what I would envision is that this will be in the brain because it is safer and it is going right to the target. And what is the target? The target is the neurons, because we know that these are the cells that are lacking these postsynaptic densities and this postsynaptic protein. And with this, we establish pretty much the same, I think, the same strategy. This synapsin promoter, which is a tag that will bring this gene to the mature neurons, okay? The human DLG4 and the poly. And we infect this to the brain, and then this goes all the way to the neuron and start the expression. Okay, this one. Okay, we'll skip this. So eventually, we want to give this in the brain. Specifically, we give this into the CSF, where the cerebral spinal fluid goes. This is the liquid that really feeds in the brain and the spinal cord, that you see here. And then when we inject the virus will go into this CSF and spread into the different cells. And because it has this synapsin, it will only get into the neurons and will not or mostly because we are using AAV9, it will love to go to the neurons and not other cells. But even if it goes to other cells, it will not be expressed. Why? Because it has this tag that is synapsin. Only neurons, I will be only expressed and produce the protein in the neurons, in the mature neurons even. Not in the neuroprogenitors, not in other types of cells. And then we will have this, our transgene, our vector, expressing in these neurons and start to look at these different outcomes. So the first thing we did, in order to make sure that the vector that we designed and we produced is functional, we tested it on mice. We took wild-type mice and injected these mice, as we will inject eventually the patients, and so that there is expression. And the PSD95, the protein, is expressed in the cortex. And mainly here we look at the cortex, but we also look at different other regions. And I want to emphasize, just to make sure, and this is for Zainab, we choose to work on both isoforms. So we have vectors from isoform A and another from isoform B, just not to start the work again and again. So it is expressing, and we are happy that it can go into the neurons. We validated that these are neurons, and everything looks in vivo in these mouse that are wild type. They are not diseased. We are checking expression, and it goes to the right place in the brain of these mice. Now we wanted to characterize them in the mutants, right? And this is where we started working on these different mutations, the different mutations I mentioned. And we started, and I'm going to show you first the 2D with the neurons, the two-dimension culture in the neurons. And you can see here, these are neurons that are being differentiated as a function of time. 4 days, 14 days, 21 days. Usually we can differentiate these into maybe 50 days. But this is a protocol that is accelerated. And we can get mature neurons by day 21 and 30. It will be really very mature. It can get even to other intervals where you get even more and more maturation. If you look across these different panels, what you see is these bodies of the cells, but then you start to see these dendrites and axons that are coming from these neurons, which is really where the PSD95 protein is expressed, where it allows these synaptic interaction. Overall, we did not see big changes at the bright field using the microscopy. You can see less, more dense, but we did not feel that at this resolution we are seeing differences or main differences. The first thing that Dania did is that she stained for PSD95. She used an antibody, what you see here in white. So these are neurons. This is the wild type, and these are the different mutants. And what you see in white is the BSD95, the protein level. What you see in red is this MAP2. Everybody knows what is MAP2 because it was explained, right? This is another neuronal marker that maps these dendrites and these axons of the neurons, not it maps the neuron basically. It's just a counter staining. And what can you see here? Anybody? If you compare the wild type to HF or frame shift or nonsense, do you see any difference in intensity or in expression of the protein? Yes, no? Yes. What do you see?
The sense it's more, like it's exaggerated. So rather
You see that it is more here?
Yeah.
And here?
It's more.
So there is not, I would say there are changes. This picture was taken by a digital scanner, and we decided because we don't see enough puncta that this is not an enough resolution. So let's go and do a higher resolution microscopy where we can see now much better. Oh, this one. You got me. And this is where Dania started working on what we called confocal super resolution microscopy. In order to see these puncta, the BSD95, what you see are these dots, and compare with the different other mutants. Now what do you see? Do you see less or more? Who see less? Less, less, less? Okay. We don't want to guess, so what Dania did is that she adopted a very sophisticated algorithm that can go and count, like AI, but now an algorithm that can count these punctas, these white dots along the MAP2. This is the marker for the neuron, and those blue dots, that is, I don't know what's the blue, but those are compare the wild type to the mutants. Okay, so the algorithm would compare, would really count those dots here, and then compare what is happening between the wild type and the mutant. And now what do you see in the mutant? Is there less or more? There is less, right? So she asked the algorithm to count. And this is what happened when there is counting. This is wild type, what you see here in the bar. And these are the different mutants showing that there is less puncta, less of this PSD95 in these 2D neurons. Okay? She looked at DLG4 mRNA expression, basically not much of a difference. The protein level is very different. Okay? Then she said, okay, what if I now bring the gene therapy? Can I restore these puncta? So you're looking at wild type, then you're looking at Harvey, then Harvey plus AAV9. You are re-expressing the PSD95 in these 2D cells. And we're looking at 30 days in vitro, like these neurons. The same for the CHIO after infection with the AAV. The same for the other CHIO and after infection with the AAV. And you can certainly see that this puncta now is restored. We are restoring the expression of PSD9 protein when we are infecting them with this gene therapy platform. Don't believe me. This is a computer. This is AI, trusted one. You can see there's a reduction, then it goes up with the AAV, goes up with the AAV, goes up with the AAV. So it's not only that we are now can conclude that these three different mutants, they are loss of function most likely, but all three, but this means that we can also correct them with the gene therapy or with the AAV. But this is only one aspect of PSD95. It's not enough for us as researchers. We want to study and to look for other things and other aspects. The other aspect that we, Dania, decided to look at is what happened to these other proteins that there was a question earlier this morning. You guys are very smart. I would like to hire all of you in my lab to do the experiment. So what happened to these other members of this post-synaptic density place, this hub that allowed the crossover of the electrical signal? What happened to them when we have deficiency or when we have these mutants in DLG4? And what happened to them when we restore by gene therapy? Okay? So we looked at AMPA receptor. You heard in the morning, AMPA receptor, NMDA receptor, right? Everybody knows. You have this in the test. Wait. So we looked specifically on AMPA receptor. This is a receptor that allows this electrical signaling. And you can see that it binds to this PDC2, and it is embedded in the cell membrane of this postsynaptic membrane, and it is helping, or the scaffold protein is just making it there very tethered to the cell membrane. So what happened to this? And this is when we stain for AMPA receptor. Okay, this is what you see now in yellow. So Daniel likes colors. So she put yellow and red. We can get here very nice. And this is what you see all in the dots, these puncta with this AMPA receptor in the wild type. Then she looked.
Puncta, define that. Is that just?
This is the structures that looks like dots. We call them puncta in microscopy. Okay? They are a cluster of proteins that are coming together and looks like aggregates.
Thank you.
This is the professional way to call it. Harvey, or this mutant, second mutant, third mutant.
Yeah, just use that.
Oh, you want me? This is too much for my brain. No.
Okay, well,
So I'll stay here. The problem is I don't have a laser pointer. So, okay. Wake up. So I'm jet lagged, you can imagine. I'm half asleep now. But what you see here, I think very nicely that if you compare the mutants, you can see much less of the staining of this AMPA receptor, and you can rescue this with AAV9. No kidding, guys. And this is the quantification. Less, more. This is the less, more, less, and more. Okay? Get me confused. So we can restore this expression of AMPA receptor at the protein level. Again, we are not, I don't know what happened at the RNA level. We didn't check. But I don't care. We really...
Westerns?
Hold on, Alex. You are an excellent student. So this is the puncta for AMPA receptors. The next question that Dania wanted to address is what happens at the synapse level, right? So these are proteins, PSD95, AMPA receptor, but at the synapse level, what happens at this cellular level? And what is at the cellular level? There is these spines that are really mediating those wiring or crossover of the signals, right? And it is quite clear to the community that if you have overexpression of PSD95, you will have more spine number because it is a scaffold protein. It stabilizes these spines. It stabilizes these structures. And the size also is increased. If you have knockdown of PSD95, then you have less in number, and the synapse number is also reduced. Okay? So let's test this now in our mutant cells and what happened after therapy. So we start with this scanner, and you can see the mutants compared to the wild types, and we look at the spines, and we're looking at these dots here, the white dots that are PSD95,
that are colocalizing with MAP2, which is the marker for the spine in this. But you can see this from the morphology of these spines. And this is what happens if we now rescue compared to the mutant. So you can certainly see there is less white puncta that is decorating the spines, whereas when you do the gene therapy or when you take this treatment with the AAV, you can have more of this white puncta. And this can be quantified, and you can see that it is less, then you increase, less, then you increase, less, then you increase, something very similar to the wild type. And this is confocal microscopy, the more resoluted, highly resoluted microscopy, where you see these puncta very, very nicely and clear that it is rescued when you compare them to the mutant and to the wild type. And this is how we train our algorithm in order to count them. So we restore the spine numbers, and we did not do a very high resolution to look for the size and the architecture, because they also have different morphology, but we are expecting that this will follow also. This is an interim summary, which I want very fast to go. How am I doing with time?
You're way over.
I'm way over.
I'm just joking.
So thank you. Bye. Going back to Jerusalem. So very quickly, we have shown that in the mutant, we have much less puncta dense levels or PSD 95 levels. We show that these levels can be rescued by the gene therapy. We saw that there is reduced AMPA receptor, so it is at the cellular level, we're also affecting other effectors, and there is also less spines, or there is spine deficits in these mutants. All of these are rescued by this AAV synapsin DLG4. We wanted to go a little bit more sophisticated at a higher resolution with organoids, where we look now more at those cell population interactions, because neurons in the brain are not found alone. They are found with other cells and surrounding them. So we did this cerebral organoid protocol of Knopfler and Lancaster. And we looked at 2 and 5 months of age organoids. Still, I want to remind everybody, brain organoids, these are resembling what happened in brain development. So these are mimicking human brain development, but they are coming from the patients. So we would like to use them because they reflect what is happening in the human brain development of these individuals or these kids. So it is better than a mouse, of course, but still they are in vitro and they have their own shortcomings. Cool. These are the, I think, the 5 months organoids. You're looking here, the wild type, we have 2 mutants. And you can certainly see that BSD95 is reduced with these 2 mutants. Okay, so this is confirming what we saw with the 2D neurons. And you can see the quantification here. I don't know why you see this, not so nice. Western, this is an important thing, Alex, right? So what you see here is looking at these 2 mutants, comparing to the wild type. This is the 95, but the protein expression is happening in two levels. There is this high molecular weight, and there is also this lower molecular weight, right? You saw this in yours also. A little nicer than yours. Little. No offense, sorry. This is not me, this is Dania. I have nothing to do with this. But look at this, Zainab. mRNA is not much of a change. I don't know. Okay, so in the discussion. Okay. So we looked at different parameters, but I think So there are differences, and there are differences in RGs in these different populations that are compromising the organoids. There are different, I would say, levels of proteins or genes that are characterizing mature early versus late. But I think what I would like you to see is this thing, where we also use the gene therapy on those organoids. So this AAV9 on this 5 months, we can infect them very nicely. And we can use Western blot, okay? And we can quantify it. So you can compare wild type to Harvey and Harvey with the fusions, AV, like this is just triplicates. And you can see that there are some differences, but all of them, they show more expression compared to the mutant or to the wild type at the same time, 180 days. Okay, so this is, I think, we'll skip. It's just saying whatever I was focusing on. I think this is very, very nice and critical. This is the third chapter I'm sharing with you. And now we're looking at the functional synapse. We're looking at the physiology of what is really happening in these mutants when we talk about electrophysiology. What is the electrical outcome or output of these mutant neurons? And what happens if we now re-express the gene using our AAV-based platform? We use for this the multiple electrode array. You heard about it, right? So this is kind of a lot of electrodes. We put on them the neurons or the organoids, and then we just record from them what electricity goes through, what is the input and the output, and what we call the local field potential, potential, and how much these neurons fire, what is the activity, and whether they have this difference in spiking and in synchronization between them, between the neurons. And let's say before this, I don't know how to go back, but red button. Oh, cool. Now I know. So let's ask the question, what would you expect from these neurons that they have less PSD95, less protein? Do you expect to see them more active or less active? Who's saying less? Who's saying more? Only Sharon says more. Okay, so I will not, because we don't have time. So what we saw is compared to the wild type is that we got less. They're less active because they have less of this scaffold protein. They do not really do enough activity of these neurons. And this thing is actually, even in the wild type, goes down with time because the neurons are somehow is now more, with time they have less activity. Maybe they are not functioning as we want them to, but relative speaking, we see that always the mutants has less until we get to this 24, 25, Sharon, where we start to see some of those mutants getting more activity, okay? So there are changes, but most of the tests that we did on these cerebral organoids is that there is less activity in those mutant organoids. So Dania wanted also to look at the neurons, the 2D, and again, I'm sorry, you don't see this very well, right? But what you see here is the 14 days, the firing, and you indeed see much less activity at 14 days. Also the spike amplitude again, which suggests the networking between the neurons is less. But when you look at 21 days, more mature neurons, you start now to see more activity of those mutant compared to those to the wild type. The spike amplitude remains less, which means that there is a problem with these mutants in the synchronization of the signal and this high level of interaction. What attracted our eyes is this Harvey, that it did not really go very high in the firing compared to the other two mutants. So we wanted to get into a higher resolution and look at this even more in details. And this is what Dania started to do with this mutation. So we're looking at the puncta, and you can see that there is less in the missense compared to the wild type of the BSD95. You look at Western, okay? So this is Western, Alex. You'll see this is the day 9, 14, and 21. So BSD95 goes up. In the Harvey, it is much less. It starts to go up day 21, very late. That meaning that the maturation kinetics is very different. And even in those lower molecular weight is also different. The same is here. This is in organ. This is in neurons. This is in organoids. There is much less in the missense in this specific mutation, and also in the organ. This is AMPA receptor, and this is BSD95. So even at these other levels, we have changes that are consistent with what we saw before. But what happens on the functional synapse? Again, the firing rate is reduced, and this is organoid data. In week 14, 15, and 16, it is less significant, so there is changes, but it is certainly much less, probably telling us that there is less activity because of the missing of the absence of BSD95. However, it is consistent that the spiking and the synchronization if this mutant is much less, regardless of the time that we looked and checked this. And what is interesting, and is,
wrap up in the next couple minutes?
yes, is that with AV, we take this back to the normal situation. The very nice and important thing that I want to close with is that we heard from Tom about sleep disturbances. And we heard about the slow waves that are characteristic of neurodevelopmental diseases. So this is what we wanted to do. So we took all the data, Dania, and looked at different levels of frequencies, okay? I want you just to pay attention to two things. This delta, which is very high frequency, or very low frequency, sorry. It is associated with this deep sleep, and it is often increased in these epileptic encephalopathies, and most likely in DLG4, okay? Whereas this gamma, which is high frequency, they are associated with high level of interaction and of thinking, and it is more sophisticated, so it is memory, it is IQ, it is, so you have more of these frequencies. What happened in our mutant is amazing. I'm so sorry you don't see this very nicely, but you have to believe me that in the mutants, we see increase of this delta, increase of these slow waves, increase of those waves and frequencies that are associated with sleep disturbances that Tom mentioned, and decreased with those that of the gamma that is associated with the high Q and this more sophisticated problems in the brain. And this is corrected. Again, you have to believe me. This is corrected with the rescue and AAV. This is reproduced in the 2N. Exactly, the delta is higher, the high frequency and the gamma is lower, and in 21 days and 16 days, exactly the same as we saw on the organoids. And I wanted to tell you, but Tom exactly explained all these, that this is what happens in the DLG4 and epileptic encephalopathies. Okay, so this is the bottom line. I think I very safely and convincingly, at least in our data, we can say that this can successfully rescue many of those phenotypes at different levels, cellular, functional, molecular, that we believe that this is promising. And this is actually the summary that Dania did, where You see very nicely, this is the wild type, this is the normal situation. You see very poor synapse here, much smaller, much less synapse and spine, and electrical activity is also impaired. And we want to believe that with this AAV platform we design, we can reverse this thing. This is the most important. This is the group. This is Dania, half asleep. This is because she doesn't sleep. She always works. And our collaborators, I'm very thankful to them. We received some funding at the beginning from Hope for Harvey, but also the Shine Syndrome. Huge thanks to you, to those that contributed the cells. Without this, we could not make any of this. And thank you for your attention, everybody. And this is Dania.
Okay. I just do mine. Okay. All right. Well done. Well done. Sorry.
We're going to do questions, right, for Dr. Rami right now.
Okay.
right.
Okay.
Okay.
Okay. All right. Real quick. It's just how heartwarming it is to see the final picture with a combination of Israeli, with Jewish and Islamic trainees with you. That really warms my
Yes,
heart to
yes.
see that this day and age. Do you have a question over there?
Yes.
Yeah. Okay.
Same question as Dr. Motri.
Okay. Say it again.
Same question.
I don't remember
The question is that how confident are you that since you saw that you rescued in all three, that all the mutants that we believe are loss of function, that the AAV9 is going to work?
So I will make a strong statement, and I think we had the discussion like 3-4 years ago, and I told you this is loss of function, and this is haploinsufficiency.
So you think that?
But I think this is the rule, which I believe. But because there are some evidence for exceptions, we have to make sure that we look at this at the functional level. Again, I would love to compare RNA with protein with function. But I would say that this would be the initial prediction. I think most of the data that also presented by the other speakers suggest that this is haploinsufficiency. There could be some mutations that need to be tested. The testing so far was only not done. I would love to see what I would believe, protein, puncta, activity by MIA or by any other electrophysiologist, just to see that this AAV would not make things worse. Mm-hmm. It could be that there is a truncation that is what you see in the splice variant that is not relevant. You bring in the wild type and you can correct the phenotype. I really don't know, it needs to be tested. But I would suggest that we look at this as loss of function unless we prove otherwise for some mutations. Just to make it easy for the community.
We think we're seeing a truncated protein in one of those mutations. And so we think that might be an example where there is a truncated protein.
With a nonsense.
Yes. We'll actually, we'll review the Westerns with you just to show. But it looks like we may have answered at least one case where there is a truncated protein that is present, but the AAV has swamped it out. Tim, as you said, swamped it out and regained function. But to discuss in the bar or over a beer. Yes, go ahead.
So the NGN2 induction process, are you using a lentivirus to deliver that and then doxycycline to drive it?
Transposons.
Okay, so there's no concern about the integration of it in terms of that?
No, we don't use lenti.
Okay. A second question that's different. Did you characterize the organoids in terms of the GABAergic composition to glutamatergic, because you're seeing high-frequency oscillations, which typically don't show up in organoid systems unless you've got these fast-spiking interneurons that are kind of troublesome to produce?
So this is a protocol that is tweaked to produce dorsal. It's mostly glutamatergic, but we always see some GABAergic. We never had, NX 2.2 is always there at a certain level, and it is variable. So EI ratio could be also here something that is to be considered. I agree.
Great. I had no idea what you just talked about, you two, but it sounded very impressive.
Okay, most technical. This is why I was fast, you know.
Okay. So I think Gerardo, no other questions. We move on to Gerardo. Gerardo, we have no time. We're just. Okay. Oh, a chat question. Okay, go ahead. Yep,
He's here
yep.
tonight over dinner.
Tomorrow morning, I'm here.
Let's address the chat questions first because they won't be here tonight. Okay, I need that.
Sorry. Oh, you had one.
Okay, so this looks like a patient question. Is there or will there be any research done on the effects of gene therapy with a splice site mutation or a gain of function mutation? It's relevant for me as well.
Yeah, I mean, happy to, if we talk, maybe if we got some lines that we can test them, maybe this will be
And
a way
resources to
get
support
some
the
resources,
testing.
because we are really deprived. Everything is done from savings. I mean, we got very little amount of funding from the foundation, and so far we didn't get any grant on this.